Welcome back. For this lesson, we're going to be focusing on mitosis, which is falling under the cell division portion of Unit 3. So we're going to be looking at 3C and keeping in mind that 3C has a big idea, the structure of DNA and how it stores genetic information for cell division, differentiation, and maintaining all organisms. In order for us to see that big picture, we're going to be focusing on two learning targets. At the end of this lesson, I'm hoping you'll be able to describe the state of the DNA and organelles in each phase of mitosis, and that you'd be able to illustrate a cell as it would appear in each of those phases. I have identified these terms that you should be keeping up with from the last lesson. So we're in about the fourth content lesson, 2 practice sessions thrown in there. But these are from the haploid and diploid lesson. And throughout this lesson, I'd like you to keep your focus on what terms we should pull out to add to this list from the mitosis lesson. Just to recap, when you're looking at a cell, what I want you to be able to do is identify from the last lesson, see how well you're feeling about this question here. Answer this question: what is true about this cell, keeping in mind what I've highlighted here for you, that this particular cell seems to have maternal and paternal chromosomes? I was practicing with my drawing tool on the PowerPoint presentation. It's not awesome. But just to recap, all cells have DNA. There are 2 types of cells, prokaryotes and eukaryotes. Prokaryotes have DNA, although they don't have a nucleus. Their DNA is shaped like a little rubber band, like a circular loop, and it's floating in the cytoplasm. Eukaryotes contain all their DNA in a nucleus. They have a lot more DNA. It's a lot more complicated. And the reason we care about this is because they're all alive and they have functions. They need to make energy, they need to reproduce, and they need to make proteins. Can't do any of that without DNA. Why do you think cells need to replicate in your body? So we've talked about this in general. I want you guys to think about it, put a response down. Maybe think about what kind of cells would need to replicate in your body or not. Since eukaryotic cells are complex, their reproduction is complex. So we talked about sexual reproduction in this case. All of these things would be part of interphase. When a cell undergoes reproduction, most of its time is spent in interphase. Their reproduction is more complicated than prokaryotic. They have to copy all of their DNA and rearrange all of their DNA, multiply some of their organelles as well, in order for that division to be complete. This is an overview of the cell cycle. We've already talked about G1S and G2 that you see here. Remember, G1 is a growth phase. G2 is the part where the DNA is replicated. I'm sorry, did I say G2? S for synthesis. G2 is another growth phase. And today what we're focusing on is this little slice of the pie called mitosis. And by default, cytokinesis, because these 2 things often go together. And there's not enough to cover cytokinesis as its own lesson. This happens to almost all of your cells. Almost every cell will go through the entire cell cycle. There are some that will not, but when they do, the result is an exact copy of the original cell. So you'd end up with 2 twins. Then that new cell, those 2 new cells would go through that cell cycle all over again. The first phase to talk about in mitosis is, this is the cell cycle, sorry. This is G1. So just to reiterate, we're talking about a rapid growth phase that the cell undergoes in order to prepare it to divide later. Everything is functioning regularly. So this is about 90% of the cell's life is going to be in some form of interphase. Remembering that interphase includes G1, the S phase, and G2. The second phase is what we call the S phase for synthesis, means to make something. In the S phase, DNA is copied. So before each cell division can occur, you need to have enough DNA to split in half and make sure each individual new cell has enough that they need for proper functioning. So the DNA is replicated or copied. The chromosome now is going to look like that X shape that we discussed earlier, and that's because what would the original chromosome, when it duplicates itself, it ends up making like a Siamese twin, and the two chromatids, the Siamese twins, for lack of a better word, are joined at one central mirror, a central spot in the middle. And the end goal is to divide into two cells. All right, and so that's why you have to duplicate all of the DNA. In order to make the DNA, the process is called DNA replication, and that's a process we're going to focus on in the next lesson. But for now, just understanding that if this is your first chromosome, when I say duplicated or the DNA is replicated, you're not going to end up with two separate chromatids like this. You're going to have two chromatids attached to each other. So although this is still one chromosome, this is also one chromosome, but the amount of DNA present is double. I'm going to wait and let that sink in for a minute. This is one chromosome, and this is one chromosome. It's just that the amount of DNA has doubled. The third phase of interphase, the last phase of interphase, the third phase of the whole cell cycle, would be G2. And that's when other preparations for the cell division are happening. The cell itself increases in volume again, and the organelles are starting to be divvied out. Things start to separate toward opposite ends of the cell. So in interphase, remember the first three phases together are called interphase. That's 90% of the cell's life. The cell will stay in interphase until it's ready to divide. So mitosis, that we're going to focus on next, is not what cells are most of their life doing, unless you're bacteria. What type of cells in your body go through the cell cycle? Body cells, gametes, both, none, or I don't know. Which phase of the cell cycle is essential for growth? This caption here is discussing cancer, and remembering from our video the other day, cancer is when they skip interphase. The cells that are cancerous, they don't go through this interphase portion. They just keep going through mitosis, and then mitosis, and then mitosis, and mitosis. It's a rapid growth. Here's a check-in. All eukaryotic cells, this is just a recap from where we are. All eukaryotic cells go through a life cycle called a cell cycle. All eukaryotic cells go through the cell cycle. It has 5 phases, but it spends 90% of its life in the first 3 phases. So remembering that's interphase, G1 for gap 1, S for synthesis, and G2 for gap 2. In G1, the cell grows and lives its life, minding its own business. In the S phase, DNA is synthesized or duplicated or replicated, depending on which term you prefer. In G2, the cell grows more and it prepares to divide. So I need you to think about how you're feeling about what we've covered so far. If you have any questions, I'd like you to write them down and bring them into class tomorrow. So here's how mitosis happens in a nutshell. You can remember this sequence, and if you can summarize this, remembering that a chromosome duplicated is just twice as much DNA, but one chromosome, as long as you can remember that, you're going to be just fine for mitosis. In the first basic part, chromosomes break out of the nucleus. All 46 chromosomes line up in the center of the cell. The duplicated chromosomes break apart and get pulled to opposite ends of the cell, and then two nuclei form around the two sets of chromosomes. Now, we're going to name all these parts here coming up, but for now, this is it in a nutshell. DNA is copied, replicated, duplicated, synthesized, whatever you want to see. See how it's one DNA chromosome here, and it's one here. It's just twice as much DNA. Then the chromosomes line up. They are divided. They are split in half. And then each new daughter cell is supposed to look like the original mother cell. So you should see two chromosomes, each with different versions of the same chromosome. So technically, the fourth phase of the cell cycle is mitosis. So that's what we're focusing on right now. There are four steps, 4 phases of mitosis. So with prophase, that's when, like I said, they bust out of the nucleus and they're sort of just floating around while things are a little bit chaotic. They're not necessarily organized in the center yet. So this is what they would look like, You'll also start to see these little fishing lines come out. These fishing lines are called spindle fibers, and spindle fibers come from these things called telomeres. There are telomeres on each side of the cell. Think of them like fishermen. So they're going to cast their lines, and they're going to start trying to find the centromeres of these chromosomes. That's prophase. In metaphase, the fishermen, these telomeres, they line up the chromosomes along the meridian or middle or middle of the cell, and they start to reel them in. So as they're shortening their own spindle fibers on each side, it ends up being a tug of war. And we call it meta because the word meta, or the root meta, refers to middle or central. Okay? So metaphase, they line up along the equator of the cell. In anaphase, the two chromosomes pull away from each other, away for anaphase. So the chromosomes are split, and the new chromosomes, now they look like chromatids, are pulled to each side, opposite sides of the cell. The spindle fibers are shortening, and the telomeres are reeling them toward themselves to the opposite side. In telophase, telo means distant. So in that case, they're as far away as they're going to get, and the nuclei, because there's 2 of them, start to reform. So just to recap, pro is kind of messy. It's chaotic. There's some fishing going on. The spindle fibers form from the telomeres, but they're not really organized quite yet. The nucleus is breaking down. Metaphase, they line up like a parade in the middle, meta for middle, and the fishermen, the telomeres, start to organize their spindle fibers. Meta, middle. In anaphase, away, the chromosomes are ripped in half, so each chromatid moves toward the telomeres, and their spindle fibers are shortening. And the chromatids are moving away in anaphase. And then telophase, the distance phase, these chromosomes are as far away from each other as they're going to be able to get, and the nuclei start to reform. We'll discuss cytokinesis here in a second. We're calling that step 5. So make sure that you're comfortable, that you can identify these phases, and that if I asked you which one do the chromosomes line up in the middle, which one are they pulled away from each other, which one is when they're the farthest away as they can get, which one is where the spindle fibers start to form, etc. So let's practice what you just learned. Identify all of the following. The cell cycle ends with 2 cells that are identical. So it happens in your body cells, not your sex cells. How many chromosomes does your body cell have? Once the cell divides into 2 cells, how many chromosomes will each of them have? And where did the extra DNA come from? I thought this was a cute little illustration just to help us get there. So that last and final phase is called cytokinesis. And in cytokinesis, the cleavage furrow begins to form, which is what you see here as this cell membrane starts to pinch together. It's not my term, not my favorite term, but that is the official term called a cleavage furrow. The cytoplasm divides, organelles are divvied up, and the cell is going to split in half. So at the end, we have 2 cells that are exactly the same. And you can see here why I chose to cover cytokinesis separately. It's a whole nother little process. So when you're counting chromosomes, keeping in mind, instead of counting chromosomes, I want you to count the centromeres instead. Before interphase, the chromosome in humans, the chromosome number is 46. So you can see here in the illustration, if you were to count all these dots, that's 16, and then another 30, that's 46. So in the key here, a chromosome with 1 chromatid and a chromosome with 2 chromatids. They still have 1 centromere, so count the centromeres. In the S phase, the amount of DNA is doubled, even though the chromosome number is still 46. So consider that the DNA is copied. It's still attached to the original chromatid at the centromere. So you're just doubling your chromatids. You're doubling your amount of DNA, but you still have 46 chromosomes. Because when those split, you want to have, after mitosis, you want each new cell to have 46 chromatids, which we're going to then call chromosomes again. So if you looked at these two and you compared them side by side, right? This is before and this is after. You want them to look the same. Here's another little pulse check for you. The original cell is diploid. Once the cell cycle is complete, what are the cells? Now this cell illustration only has 1 set of chromosomes. Okay, at this point, I want you to take a moment and decide how you're feeling about the 2 learning targets we've got for this lesson, describing the state of DNA and organelles in each phase of mitosis, and then whether or not you could illustrate those. So when you're trying to remember this, I want you to remember it from this kind of picture, going back here. This is the kind of picture you're going to get. Okay, this one or something like this. There are so many pictures on the Google machine. If you feel like going to visit those, you're welcome to do that. You can rewind this and watch it again, however you feel like. Come into class with questions. I'm looking forward to talking to you guys. Bye.